follow-up to the video on cellular exchange, right? We talked about our fluid compartments. We actually have all of them here, right? Here's our ISF, right? The interstitial fluid. So anywhere out here would be the interstitial space. We have plasma. I could label that here. And those two added together are extracellular fluids, right? And then, of course, we have these cells, which I've left fairly sparse. But the cells of any given tissue, which are surrounded by these capillaries I've got shown here, sort of top and bottom. And those contain cytoplasm, or ICF, intracellular fluid, right? And we talked previously about the fluid exchanges that occur across the cell membrane, right? We talked about how the cell membrane has these embedded proteins that can be pumps, like the sodium-potassium pump, or channels, like a potassium leak channel, and they do allow solutes to move back and forth. And there are transport proteins to move other solutes like glucose and other nutrients, amino acids, into the cell, wastes out of the cell, right? So lots going on there in the membrane. But fluid movement is dominated by what I've drawn here, right? Which is an aquaporin, right? And so fluid exchange between the ICF and the ISF, right, between inside and outside the cell, occurs through the aquaporins, and it occurs only in response to osmotic forces, right? We talked about that. Remember the examples of adding more, there's already solutes out here, but adding more solutes to the outside of the cell upsets the osmotic equilibrium, the normal status, the osmotic equilibrium between inside and outside. And in this scenario, we would be drawing water out of the cell across the aquaporins, right, with more adding solute to the extracellular fluid. And again, you could go the reverse and drink water and dilute the extracellular fluid and then have water move into the cell, right? Remember the hypo and hypertonic scenarios we discussed previously. Okay, so that makes fluid exchanges across the cell membrane fairly simplistic, right? They're all based upon osmotic pressure differences. Now, what I want to talk about next is the other sort of category of fluid compartment exchanges, and that's between the plasma and the ISF. So these arrows here, indicating these pressure differences across the cell membrane that result in fluid shifting. And the problem here that makes it a little bit more complicated, and we'll go into greater detail when we talk about the cardiovascular system and we come back to blood vessels, but I do want to talk about, we're talking about fluid compartments, and I want to talk about the exchange between the plasma and the interstitial fluid because it is critical. And really, solutes can be exchanged back and forth across the capillary wall by diffusion, right? We already talked about that, so we could talk about, just to draw an arrow down here somewhere, right? We could talk about the movement of, let's say, glucose or oxygen, right? Oxygen diffusing out here, right? And that's going to be based upon J diff, right? Remember our J diff equation? J for flux, diff for diffusion, right? Driven by the gradient and affected by the distance and other factors, the diffusion coefficient, right? All going to affect the diffusion of oxygen out of the capillaries. We could talk about diffusion of carbon dioxide, again, produced by the cells out here of the tissues, right? We could talk about carbon dioxide diffusing in the opposite direction because its gradient is flipped of that of oxygen, right? It's higher on the outside and lower on the inside. Right? So that's one form of capillary exchange driven by the concentration gradient of particular substances, right? And again, described by the diffusion equation, which we've discussed previously. But we also have what is called bulk flux. And bulk flux is the movement of a solution, essentially, right? So we'd basically be talking about something called J, again, for flux, and again, flux just means the movement of something. But so now we'll call it J bulk, right? And J bulk is bulk flux, is driven by not just osmotic pressures, but also by hydrostatic pressures. So now, right? So again, go back to the cell here. We had talked about how movement of fluid is only driven by osmotic forces. Well, now we've got movement of fluid driven by osmotic forces and what are called hydrostatic forces. And the other difference here is that the only thing that goes through aquaporins is water, right? They're called aquaporins, water pores, right? Whereas in capillaries, we have these gaps between the cells here, right? Where I've got these arrows sort of passing through the membrane. And meaning that we've got bigger gaps in the capillary wall. And so when water moves, the stuff in water goes with it, most of it, right? We'll come back to that in just a moment. But almost all of the solutes that are dissolved in the plasma water can go back and forth across these pores. And so whenever we have an osmotic or this hydrostatic pressure gradient, and we have both, we have to talk about which one's winning. But what that means is that those forces can drive a solution of water and electrolytes and nutrients back and forth across the capillary wall, right? So we've got this exchange between, inside and outside the capillaries driven by these forces, right? And so J bulk, right, is driven by osmotic pressure, abbreviate by OP, and hydrostatic pressure, abbreviate with HP. Right? So let's talk about them one by one. So the osmotic pressure. So what's the osmotic pressure gradient here? Well, we just said that capillaries are permeable to almost everything, right? Well, the one thing they're not permeable to is proteins. And again, this is most capillaries, right? We're overgeneralizing here. There are some capillaries that are permeable to proteins too, in which case everything goes back and forth. But for the moment, let's talk about the majority of capillaries. And proteins, these green dots I'm drawing here, right? That's a protein, are trapped, right? And which means, and we used the term before, reflected, right? So these things bounce off the inside wall and they remain trapped in here. Well, because there are so few, there are proteins out here, right? But they're sort of few and far between. Whereas in here, the density is much higher, right? The plasma protein content is much higher, which means we have a solute difference, right? Remember, the sodium can move back and forth, the chloride can move back and forth, glucose can move back and forth. It might be driven by diffusion, but the diffusion will equilibrate the concentrations, right? There won't be a gradient, right, over time. Well, there won't be a dramatic gradient over time, right? And so because those solutes can move readily, right, you can't maintain a strong gradient because it keeps moving, right? And so the only solute that really maintains a continuous gradient are the proteins. And we call those oncotic solutes, right? So sometimes this O, instead of being osmotic, we call it oncotic because that's referring to and actually it's right here, that term right there, blood oncotic pressure, right? And so those proteins are tending to drive, right? Put a color on here, drive water movement and the solutes that are dissolved in it into the capillary. Okay, so that means that based upon that pressure alone, capillaries would tend to suck up water continuously and blood volume would go up. Okay, well, that's not actually what happens. So, and that's the reason that's not what happens, is that we have also a hydrostatic pressure. And the hydrostatic pressure is simply what we refer to as our blood pressure, right? There's pressure in our blood vessels that pushes the blood along those pathways. And so it tends to not only push fluid flow, right, along the vessel, down the pipe, right, and out the other side, but it also tends to push on the walls, right? And so it actually ends up pushing outward. And the magnitude of the size of the arrows here is an indicator of how big those pressures are. And so, as you can see here, at the beginning of the capillary, the hydrostatic pressure is dramatically dominant, right? Which means you've got an inward force, but you have a larger outward force, which means your bulk flux would be outward. Now, one of the things that happens is that as fluid flows through a pipe, the fluid pressure falls. And that's due to friction and other things that we'll talk about when we get to the cardiovascular chapter. But notice the size of these arrows, right? They keep getting, the outward ones keep getting smaller, right? And again, we still have the inward one, right? We still have the proteins trapped here, but the outward one is falling. And so, this outward one is driving what's called filtration. Oops. Right? Moving fluid and small solutes out, but leaving the proteins trapped behind. They're being filtered out of the plasma, remaining behind. And then the inward force is driving a process called reabsorption. So you've got both going on simultaneously, but look who's winning over the length of the entire capillary here, right? At the end, it might be a bit of a tie, right? About the end of the capillary, it's sort of like, okay, now they're both about equal, and the inward and the outward fluxes cancel each other out. Okay, but if we add up what happens across the whole length of the capillary, we get filtration, right? Now, notice we have two capillaries here, though, right? We have this capillary on the bottom. And here's the weird bit about capillaries, right? Your capillaries can hold approximately, if you use the dimensions and multiply it by the number of capillaries, can hold several liters of blood. Okay, your total blood volume is between 4 and 5 liters. And the capillaries have the potential to hold probably more than 50% of that. But at a given moment, the capillaries are holding about 250 milliliters, right? Not even close to a liter. So what does that mean? Well, what it means is that the capillaries are not all perfused. There's not blood flowing through all of your capillaries all of the time. In fact, if you do a calculation, it's probably about 1 in 10, 1 in 11 capillaries that are being perfused, and the other, so here's one being perfused at the top, perfused meaning blood is flowing through it, right? But at the bottom here, we have an unperfused capillary. There's no flow, right? There's no inward arrow here. The blood is just sort of sitting in it, right? Not moving, which means the hydrostatic pressure inside, and the reason that's true is that the vessel earlier upstream of this is constricted. It's pinched tighter, so that there's no blood flow through this capillary at this moment. Now, again, that's not going to remain that way, right? We talked about autoregulation and how blood sort of can shift from one area to another, blood flow, based upon sort of opening and closing these vessels upstream of the capillaries. Okay, but at this moment, the upstream vessel is constricted, so there's no flow in this guy. Well, guess what? That means the hydrostatic pressure down here is really low. Note the outward directed arrow here is very small, right? Smaller than the inward oncotic pressure due to the proteins, right? We've got to have our proteins down here, right? They're still there because the blood just sort of ceased to move when the vessel constricted. There are the proteins, and they're, sucking water into the capillary, and there's much smaller outward hydrostatic pressure. So guess what? This capillary is reabsorbing. So we've got one capillary filtering, right? So perfused capillaries filter, and unperfused capillaries reabsorb. And so that's why we have these arrows, right? Sort of moving fluid from the capillary that's filtering, to the ones that are reabsorbing, right? And so we've got this net movement of fluid through the interstitial space and into the unperfused capillary, right? And so it's going to pull up some fluid over time, right? And I show it sort of skinnier because it's not being pressed open by the hydrostatic pressure. Yes, it's filling with more fluid over time, but it's not. It doesn't have an inward pressure that's very large, and so it's going to look a little skinnier. Now, not all of the fluid filtered by these perfused capillaries is taken up by the unperfused capillaries. Some of it, as you can see here, will end up being taken up into what are called lymph vessels. So these are blind-ended vessels. They're about the size of capillaries. And what they do is they collect the excess interstitial fluid. Right? So this is, right, as we filter, we're producing interstitial fluid. Some of that interstitial fluid goes right back to becoming part of the plasma, right? These green arrows coming down here, right? That went from plasma to interstitial fluid, right back to plasma. Whereas this arrow is showing the excess filtration, excess interstitial fluid is going to become lymph, right? So this is now called lymph. And this is going to, these vessels are going to travel and along the way, they'll, you know, this vessel's going to keep going, right? I'll show this guy travel along. And what's going to happen is it's going to come across these nodules called lymph nodes, right? And it keeps going and so on, right? So this is a node. And this is where we find the B and T cells that you'll learn about in the immunity section. And when we have an infection, let's say we had some bacteria, you know, due to introduced because we got a splinter or a cut, right? We'd have all these bacteria that sort of deciding to take up camp in the interstitial space, but those would end up being taken up here too, and would get sort of removed by the B and T cells that we would find in these lymph nodes. There's a B or T cell sitting in here, right? And actually removing those, they're called foreign antigens, right? Infectious elements. Okay, but so the whole idea here is that we have filtration in, right? So our J bulk, right? There's J bulk, right? It's driven by the balance between the osmotic pressure and the hydrostatic pressure. Depending upon which one of those two wins, we're either going to get filtration in a perfused capillary at the top here, or we're going to get reabsorption in the unperfused capillaries. Any excess filtration by the filtering capillaries is going to be taken up as lymph, which is passed through the lymph nodes and is eventually going to return to, right, returns to veins, right? These vessels, and again, in the anatomy portion of the course, you'll learn about the lymphatic anatomy, and the lymphatic vessels return this fluid, cleansed fluid, if you will, to the veins. So in reality, right, so our circulation here, and it's very useful to, again, for this immune function that we're talking about. It's also essential for sort of picking up interstitial solutes and returning them to the bloodstream, right? Let's say this cell is secreting a protein, right, of some sort. Well, now it can end up in the bloodstream, right? You know, if it's a hormone or some other molecule that needs to end up in the bloodstream. But so our fluid movement here is from plasma to interstitial fluid to lymph and back to plasma, right? So it's a circuitous pathway of fluid movement that's really important. And it's very significant because the amount of lymph produced per day exceeds the amount of plasma we actually have. In other words, the entire plasma volume leaves the bloodstream about twice a day, maybe even 3 or 4 times a day, to, you know, follow this sort of circuit from plasma to interstitial fluid to lymph. So it means your plasma is cleansed multiple times a day, right? Well, not plasma per se, but the interstitial fluid produced from the plasma due to filtration is cleansed multiple times a day. Okay, and that is capillary exchange driven by oncotic pressure. Remember, that's an osmotic pressure, right? But the term oncotic refers to the proteins. And balanced to some degree by the hydrostatic pressure, but not perfectly balanced, as we can see here, right? And so we end up with filtering capillaries and reabsorbing capillaries, and the excess becoming lymph.